everyone's quiet. All right. First of all, good morning. All right, here's public service announcement. Anyone sitting back in this area by the camera, please do not talk loud or put your cameras out during the talk, because if you do, the feed will pick you up versus up here, so we really want to hear the speakers. So that's, if you're in that area here, just kind of be as quiet as you can, so to speak. All right, my name is Brad Caskey. I'm the Provost and Vice President of Academic Affairs at Birmingham Southern College. It's my distinct pleasure to welcome you to today's Provost Forum. Today's speaker is Dr. David Schedler, Professor of Chemistry, who joined Birmingham Southern College in 1994. Ouch! Nice! His academic credentials include a BS from Rhodes College and a PhD from the University of Alabama. Do I have to say Roll Tide? Okay, I won't say it to you. I'm still learning the Southern culture, what's appropriate and not. A few of Dr. Schedler's areas of interest include enzyme inhibition and new synthetic methodology. The title of today's provocative presentation is Halogenated Aminoglycosides, a new approach to overcoming antibiotic resistance. Please join me in welcoming Dr. Schedler to the podium.
All right, well, thank you, Brad, for that introduction. And as you might have noticed, the title has changed. But thank all of you for being here. And in fact, I have to apologize for the title. I tried to come up with something a little sexier, but what can you do? So, ignoring the title, I wanted to start by introducing you to this molecule that we're going to talk about, Kanamycin-A. Kanamycin-A is in a class of antibiotics known as aminoglycosides. And basically it has 3 sugar rings that each contain an amino group. That's the NH2 group. And then they're connected by a glycosidic bond. And so that is why it has the name aminoglycoside. And as we find out later, you'll see that we're going to do modifications, as the title says, to the 6th position, that is this position on the ring. But before we start on that, I want to give a brief introduction that kind of encompasses everything, and then we'll kind of break it down. Because in general, my work involved the investigation of the binding affinities of modified Kanamycin-A's to the enzymes that are excreted by bacteria as a natural defense. So these enzymes are excreted and they bind to the antibiotic and destroy it. And so that is the main topic of this work. And so this ability to destroy the antibiotics by the excreting enzymes is one of the things that leads to antibiotic resistance. And I know there's a lot in what I just said there, but I can illustrate that a little better on this next slide. So if you have an antibiotic that you're using to treat bacteria have developed enzymes that they can excrete, and the analogy I'll be using is it's like a missile defense system. That enzyme will actually go out and intercept the antibiotic, bind to it, and destroy it. And so that then would lead to resistance of that bacterium to that antibiotic. And so what we're going to do now in the discussion, I'm going to kind of start general, and then we'll get more specific. And I'll use some examples until we get to the exact work that I, in fact, did. But I'd like to start with some acknowledgments. First, of course, Birmingham Southern College and the development committee that shepherds the sabbatical program, which is very important to our research, and also to Linda and Brad and Tim for their support of the program. And also my colleagues in chemistry and physics that are listed there, because all of them made sacrifices to allow me to do the sabbatical and do my work. And then, of course, Isa Delgado, who nothing happens on the 3rd floor of the science building without Isa Delgado. You need anything, she gets it for you. She knows where everything is. So thank you, Isa. I'd also like to acknowledge my collaborators on this project. This started as a NIH grant that actually ended up not being funded, but we still started doing the work. And all of these collaborators are at the University of Tennessee, David C. Baker, Anjan Sapertsu, and Adrian Norris. David Baker is a chemist. The other two are biochemists, and we'll see some of the biological testing that they did. And I'd also like to thank two undergraduates here, Sarah Gwen Moore and Sarah Bowman, who kind of took over this project and did it as their senior research this past fall. I'd also like to make a couple just quick personal acknowledgments because they're actually here. My children, Margaret and Lucas, for their support, and Andrea Lee Bailey, my partner that's here, and Lynn Adams, because she volunteered to bring the kids here. But I would be remiss at this point not to acknowledge someone else. One of the things one of the contributing factors that makes any sabbatical successful is to have a professor that takes over for you in the classroom. And without the peace of mind offered by having a skilled and knowledgeable colleague, I would not have been comfortable leaving my students and really focusing on my work. And Craig LeGrone was that colleague. And unfortunately, we lost him this January. He'll be missed. And so I'll begin with an outline of the talk. I'm going to talk in a minute about what the actual sabbatical goals were, so you can see how far short of that I fell. And then I'm going to do a general discussion of antibiotics and then antibiotic resistance using kind of a common antibiotic to illustrate examples of this. And then we'll talk about how we use these mechanisms of resistance to our advantage to come up with chemical strategies to then allow a chemical synthesis of analogs that could overcome the antibiotic resistance. And then I'll have, of course, conclusions at the end. Okay, but before I actually get to the goals, one of the reasons I chose to do this project is when I was a young Turk chemist in graduate school, I worked with fluorinated cyclotols, which are halogenated sugars. And, as a young Turk chemist, I guess now it makes me an old turkey. So we basically made these halogenated sugars and we studied how they were binding to enzymes in different metabolic pathways. So this experience kind of informed this project. And so this project has kind of allowed me to get back to my chemical roots. And so what were the goals of my sabbatical? I'm not going to read the whole thing off the slide, but basically what we're doing is we're trying to find aminoglycoside antibiotics that we can modify that will not bind to the enzymes that have evolved to destroy them. So we're trying to come up with antibiotics that missile will not intercept and destroy. And this would allow those new aminoglycoside antibiotics to still be active against the target bacteria. And a goal in any sabbatical that we do is to introduce a new research opportunity for students. And in fact, that's already happened because, as you saw, two of my students have already started working on this project. And then another goal was to have them present this work at a national or regional meeting. Okay, so before we go forward, who are the players in this study? The first is bacteria. And bacteria are unicellular microbes that cause disease in humans and animals. But not all bacteria are bad. As we know, we have a symbiotic relationship with bacteria. We have them in our gut that help with digestion. But we're casting for this performance the villains, the bacteria that actually do cause disease. The second player are enzymes. And these are the analogy I use. This is going to be the missile. And enzymes basically are proteins. And in this image, I'm showing in teal the actual structure of the enzyme. And then this red, white, and blue piece is actually a smaller organic molecule that is bound into the active site of that enzyme, because enzymes are proteins that bind molecules and do some chemical transformation to them. And then also the last player are antibiotics. And those are toxic substances that are released by bacteria to actually kill other bacteria that are competing with them. And so really the central player in this is the enzyme. That enzyme is the missile in our analogy. And what it's actually bonding to here is an antibiotic. And then it transforms that into something that's inactive. And that is what causes the the resistance. So what we're looking at, basically, I hope in this general discussion, is how these three players interact to make antibiotic resistance possible. Okay, so to begin, let's talk about bacteria. The whole premise of this project starts with the fact that bacteria have to aggressively compete to survive. And in order to do that, they release toxic substances which are called antibiotics. So the important thing is antibiotics are natural products. They're actually produced by the bacteria. And you can see in this table, again, I'm not going to go through every one, but just some examples that you might be familiar with, like neomycin and streptomycin are both antibiotics that are released by these strains of bacteria. Sarah Gwen, I'm doing pretty good so far. And then there are other microbes, too, that release these antibiotics, like the mold Penicillium, which releases penicillin, and that's going to be something that we revisit a little bit. And you know, when chemists and biologists first started looking for treatments for bacterial infections, they looked to chemical methods. For example, sulfa drugs, and the German physician Ulrich looked at arsenic compounds that actually killed the bacteria that caused syphilis. It wasn't until the British scientist Alexander Fleming discovered penicillin that we actually had the knowledge to take advantage of what bacteria are actually producing to use to treat infectious disease. And what Fleming noticed is that the fungus Penicillium would actually kill Staphylococcus aureus, which is the bacteria that causes staph infections. And it's kind of an interesting story with penicillin. It was discovered by Fleming, and the story goes that he was growing colonies of Staphylococcus aureus in agar and petri dishes, and he went away on vacation. And when he came back, some of those places had been infected by a mold called Penicillium. And what he noticed is there was a zone of inhibited growth around those colonies of mold. And so he hypothesized that there must be something excreted by that mold that is killing the bacteria. He also hypothesized that could in fact maybe be isolated and used medicinally for infection. But he didn't have the facilities in his lab to really do anything with that. And so he wrote a paper and, just forgot about it. Well, about 10 years later, and, in science there's a lot of serendipitous things that happen. Two chemists at Oxford, or actually I said chemists, two scientists at Oxford, were actually working with molds and bacteria, and they were looking through some old back issues of the British Journal of Experimental Pathology. And they just happened to run across Fleming's paper. And they thought, oh, that would be interesting to kind of look at the science behind that. They had the proper facilities. So then they were able to culture the mold and actually extract the substance that was killing the bacteria called penicillin. Not only could they extract it, but they were able to purify it. And they were able to test it and see that in fact it did kill S. aureus. But they wanted to test it medicinally. And so they found a case, Albert Alexander, in 1940. He actually was pruning his rose bushes and cut himself on one of the thorns. And he developed a staph infection that quickly spread to his eyes and his scalp. And so Florey and Chain came in and gave him some of that extract, and it actually was working to cure the infection, but they ran out. And so unfortunately then Albert Alexander ended up dying of that infection. Well, about 2 years later, they had in the meantime found a different source for penicillin. It's still a penicillium mold, but it was from, I think, cantaloupe, if I'm right. Something that produced far more penicillin. And so they had a larger batch of the extract, and they were able to treat an American, Ann Miller, who had a miscarriage and developed a septic infection. And they were able to treat her successfully. And so that is how penicillin came about to be used medicinally. And I'm sorry this is a chemistry talk, so I have to show structure. So this is the structure of penicillin. I just want to point out one thing about the structure. This functionality right here that I'm circling, an oxygen doubly bound to a carbon, singly bound to a nitrogen, that's called a, organic students, amide. And when it's in a ring, it's actually called a lactam. So penicillin is in the category of antibiotics known as beta-lactams. And I want to use this penicillin as an example as we go through our general discussion because it's something all of us are familiar with. But penicillin got a lot of use after the Florey chain experiment. And early on it was used for strep infections, staph infections, against the bacteria Listeria, which caused meningitis. And so a question we can ask at this point now, since this is a science talk, how do antibiotics actually work? And we'll start with just talking about the mechanism. There are actually 5 targets for antibiotics when they attack a bacterial cell. They can disrupt the cell wall, they can disrupt the membranes, they can inhibit protein synthesis, they can inhibit the genetic machinery, the DNA, RNA process, and they can disrupt metabolism. Okay, but the 2 that we want to really focus on. The first is disrupting the cell wall, because that's where beta-lactams work, which is what penicillin is. We'll look at that example. But also later on, the aminoglycoside antibiotics actually act by binding to the ribosome and preventing protein synthesis. Okay, so how does penicillin work? Okay, this is S. aureus that we've been talking about. It's a bacteria. It has a cell wall that's made up of peptidoglycan. And when it's exposed to penicillin, the penicillin inhibits this production of the peptidoglycan, and it causes weak points in the cell wall. When exposed to a hypertonic environment, you start to see water that's actually going through this weakened cell wall. And when it does, it builds up pressure in the cell and it lyses, it breaks open. And that kills the bacteria. Okay? And so, again, this is a chemistry talk. I'm sorry, so I have to show a few structural things. The peptidoglycan is actually made up, it's a lattice of polysaccharide chains. And these are acetylated sugars, abbreviated NAG and NAM. And they're connected by these small peptide chains that cross-link the polysaccharide chains. And just so we have some terminology before I move on, I'm using the word peptide just to make sure we know peptides are made by the building blocks amino acids. Amino acids are small organic molecules that are bound together in a certain order that give certain peptides. Those peptides then can interact with each other or with themselves in a 3-dimensional way to form a protein. Okay? And so how does penicillin chemically kill bacteria? Well, as I said, in order to make a peptide, you have to piece together the building blocks. So these are just 2 amino acids that are being bound together to build up a peptide. And they form, believe it or not, what's called a peptide bond. And if you notice what's in this blue square, does anybody recognize where you've seen that before? That is the amide functionality that we saw in penicillin. Okay? And so it is in this way that penicillin works. Because what it does when it gets into the bacterial cell, it binds into the enzyme transpeptidase, and that is the enzyme that actually pieces the amino acids together to form the peptide. And what penicillin does when it gets into the enzyme, remember enzymes are proteins that do transformations on organic molecules, so it actually reacts with the penicillin. It forms a covalent bond, and then that terminates the activity of the transpeptidase. It can no longer produce peptides, and so then that affects the production of the peptidoglycases. And that leads to the weakening of the cell wall. Okay? So now we want to transition. That's how antibiotics work. But that brings us to this explanation of antibiotic resistance. And it was Fleming himself that actually kind of first talked about this in his 1945 Nobel Prize lecture. And by the way, he won the Nobel Prize for penicillin, but Florey and Kane also did. So it's not one of those cases where somebody got left out. But what he basically, and I'm not going to read this, but what he basically saw is that if you treat bacteria with concentrations of antibiotics that are too low to kill all the bacteria, you can lead to something called resistance, where the antibiotic is no longer active against the bacteria. Okay, and so, a simple way of saying that is in the same way we saw that antibiotics have a target to kill the bacterial cell, now we see that actually bacteria have ways of protecting themselves against the antibiotic. So it's kind of a battle that's going on between those two things. And from an evolutionary perspective, bacteria have been developing resistance to antibiotics from their beginning. We're only in the game recently. So bacteria are much better at combating antibiotics. Okay, and that's the challenge that we face medically. So how do bacteria build up this resistance? Well, it's genetics. Bacteria lose their sensitivity to an antibiotic by expressing genes that allow new generations to stop its action. And I'll let you read a comic which kind of demonstrates that. And how do they develop this resistance? How can bacteria get to the point where antibiotics don't affect them? All right, well, let's look at this cartoon first, and then we'll get a little more scientific. If you have a bacterium and you treat it with an antibiotic, hopefully you cause death. But as bacteria are multiplying and making new generations, you will have mutations. And if you create a mutant that is resistant to the antibiotic, then that mutant and they use the term offspring here, which sounds very unscientific, but we'll use it anyway. They produce offspring that resist the antibiotics, and what you do is you end up, exponentially multiplying those mutants that are resistant to the antibiotic. Okay, a little more scientifically on this slide. If you have a population of bacteria that are non-resistant to the antibiotic, you may have in that population a couple bacteria that are resistant. If treated with an antibiotic, you will kill off most of the non-resistant bacteria, leaving your mutants. And then as subsequent populations grow, you end up with a population that's mainly resistant bacteria. Okay? Is that a serious problem? Well, if you look at this table, again, I won't read all of them, but some of these antibiotics are very familiar to us. Of course, we've already looked at penicillin, but things like tetracycline, vancomycin, methicillin, ampicillin, these are all antibiotics that we've probably heard of or taken. And this column shows the years that they were deployed, and then this is the years that we actually started seeing resistance to them. So this is a very serious issue. And from a public health point of view, what is it that's leading to this resistance? Well, there's several things. The first is the overprescribing of antibiotics. All of us, you go to the doctor and you complain about any kind of sore throat or whatever, what are they going to do? Give you an antibiotic. The other problem is that patients sometimes don't finish the whole prescription, so they're leaving strains of bacteria that can then continue to mutate and build up resistance. Also, the overuse of antibiotics in livestock and fish farming. Poor infection control in hospitals, lack of hygiene, all of these play a part. But this last one is where I come in, and that's the lack of new antibiotics being developed. This is an area that I have 2 projects in now, and that is trying to look at ways of making antibiotics that are not resistant, antibiotics that bacteria are not resistant to. Okay? How bad is this overprescribing of antibiotics? I just wanted to show you this quickly because it's kind of a staggering number. Each year, 269.4 million antibiotic prescriptions are written in the United States. And that's actually a 2015 number. And you can see even in certain states, like Alabama, for each population of 1,000, there are over 1,000 prescriptions written. So that seems like a pretty serious problem. Okay, and what does it do? It causes these things called superbugs that are resistant to antibiotic treatment. And in fact, the Centers for Disease Control estimate that about 23,000 people die every year in the U.S. from an infection from a bacteria that's resistant. Okay, so how do we combat this? Well, to combat it, we have to know the mechanism by which the bacteria are able to destroy the antibiotic. And so these are the different ways that can happen. The first is that, these are the mutants. A mutant is developed that has denied access to the antibiotic, meaning the antibiotic can no longer get into the bacterial cell. This is the most important one to us, and that is that bacteria can produce enzymes that modify or break apart the antibiotic so it's no longer effective. You can have altered target sites, so if the antibiotic gets into the cell, it's no longer able to bind where it needs to cause death. Also, a mutant can develop the ability to pump an antibiotic out of the cell faster than it can come in. And then, I guess this one you call a dummy target. You have a target that will bind to the antibiotic but then has no effect on the bacteria. Okay? So let's look at our example of penicillin, to talk about resistance. And just quickly, this is kind of the evolution of drug resistance. This is something that you've probably seen before, and that is, we started talking about S. aureus and the use of penicillin. Quickly developed this penicillin-resistant S. aureus. Then had to come up with another antibiotic, methicillin. After a while, S. aureus became resistant to methicillin, and we formed this methicillin-resistant S. aureus, abbreviated MRSA. And MRSA now has kind of become a generic term, but initially it meant, just methicillin-resistant. Now you use it for just about any resistant bacteria. And then vancomycin was developed, and it's a far different antibiotic than these two structurally, but then very quickly, again, we saw different varieties of the bacteria that were resistant. So it's a constant battle to come up with new drugs. So again, this is chemistry, so let's look at what's actually chemically happening to allow S. aureus to be resistant to penicillin. And this is an example that leads to, this is one of those missiles. We can call them an antibiotic destroying missile. There's an enzyme excreted by S. aureus called beta-lactamase. And beta-lactamase basically binds to and reacts with penicillin, breaking apart that lactam ring. Remember, that functionality was what was important to be able to enter the transpeptidase. So this new modified penicillin cannot bind into the transpeptidase, and so it doesn't have the effect of preventing peptide synthesis. Okay? And so that allows me now to transition into my work. Okay? So I'm going to use the same kind of illustration I did at the beginning. Okay? And remember, what I was working with is Kanamycin-A. And Kanamycin-A, bacteria have developed a resistance to that. So what we wanted to do is take that Kanamycin-A and modify it in such a way so when we used it to treat bacteria, when the bacteria fired the missile wouldn't intercept the antibiotic. So in other words, something that would not bind any longer to the missile. And so that antibiotic would then be able to get around the defense system, get into the bacterial cell, and hopefully then be able to do the biological effect to kill the bacteria. So that's kind of, in a nutshell, the gist of what this study is. Okay, so let's look. Aminoglycoside antibiotics. On the title slide, I kind of introduced you to kanamycin, because that's the one that I'm most interested in, but just so you know, there are others in that class. Two examples are streptomycin and neomycin. Again, you may have heard of these antibiotics. These are all aminoglycoside antibiotics, and they're all formed by some variety of streptomyces. So these are antibiotics that are natural products produced by bacteria. Okay, but Kanamycin-A is what we're interested in. And it was discovered in 1957, and it's produced, as I said, by a streptomyces variant. I'm not even going to try to say that word, as a natural defense. It's no longer prescribed in the United States due to serious side effects, but research on kanamycin may be transferred to other aminoglycoside antibiotics with less severe side effects. And these aminoglycoside antibiotics are important because they're one of the principal agents in combating these bacteria that are used in bioterrorism, anthrax and the plague. And so developing new aminoglycoside antibiotics that aren't resistant could be very useful. Okay? And so how does kanamycin work to kill a bacterial cell? And this is where the biology police might have to chime in, because I'm going to try to go through this. Inside the bacterial cell there's a ribosome, and it's basically involved in taking the messenger RNA that's coded with these codons for transfer RNA, which has the amino acids attached to them. And so basically what messenger RNA is tries to line up these amino acids in the proper order to then be formed into a peptide, which eventually forms a protein. But what an aminoglycoside antibiotic does is it actually binds into this 30S subunit, and it interferes with the codons. And so it causes mismatches and sometimes termination. And so the bacteria is not able to produce these peptides that then go on to form proteins. And so that's chemically how the aminoglycoside works. Okay? And then the next player is that missile defense enzyme, the analogy we're using. What's happened is the bacteria that we use aminoglycoside against has developed these enzymes called aminoglycoside modifying enzymes. And basically, absent the enzyme, your antibiotic binds to the proper ribosome target and kills the bacteria. But in the presence of the missile defense enzyme, that enzyme will bind to the antibiotic and do a chemical transformation. Okay? And that can lead to a couple things. Preventing the antibiotic from being transported across the membrane into the bacterial cell, but it can also prevent it from actually binding to the ribosome. Either way, it makes the antibiotic ineffective. And there are a number of different aminoglycoside modifying enzymes. So that's what makes it so difficult. Because if you find a solution for one of them, there are many more. But the common way that they modify the antibiotic is by putting acetyl groups on the nitrogens. Remember those amino groups on the sugars, and also adding nucleotides or phosphorylating the hydroxy groups. Those are three common ways that it modifies the aminoglycoside antibiotic, making it ineffective. And I can kind of show you structurally, there are several sites on Kanamycin-A where these modifications occur. And it's important that we know where those are. So you can see, at certain sites you can have phosphorylation, acetylation, or adding the nucleotides to the oxygen. All of those things end up making the antibiotic ineffective. And just to illustrate, with Kanamycin-A, if you treat it with one of those enzymes that phosphorylates, what I'm illustrating here is that if it phosphorylates at one of these positions, you totally change its ability to bind to the ribosome. And so it's no longer effective. And it's a simple modification, but it's effective. Okay? And so before we move forward, we need to really examine this binding that's occurring. Because remember, what we're trying to do is design analogs that will not bind to those antibiotic-destroying enzymes. And so an enzyme is a protein, and demonstrated over here, you see this white portion, most of the protein structure is really just scaffolding to position the active site. And in an enzyme, the active site is made up of a binding site and a catalytic site. The binding site is where the substrate, in this talk, the antibiotic, actually gets bound into that enzyme, and it orients it in such a way that the catalytic site is then able to do the chemistry on it. If you looked at that last example, this would be where the phosphorylation would occur. Okay? And so in order to start designing antibiotics that will not bind to these enzymes, what do we have to know? We have to know something about the architecture of that binding site, the interaction between the antibiotic and the protein. Okay? And so to do that, we have to know a little bit about that binding event. And the substrate here in our context is the antibiotic. When it binds into the enzyme, there are several things that are important for it to bind properly. The first is simple geometry, its shape. Okay, the next is intermolecular interactions. Things like electrostatic interactions, hydrogen bonding, all those things when the substrate fits into the site, those are interactions that hold it in place. And so what we're trying to do is know the architecture of the binding site and make modifications that will prevent the antibiotic from binding. Okay? And so going to a more specific, and by the way, does anybody know what movie this comes from? It's a James Bond movie, so you're close. We only live twice. So this is my new analogy for the missile. Because what's important, the missile doesn't just shoot out and bust up the antibiotic, it has to bind to it first. So the missile actually opens up, it has a binding site, and it binds to the antibiotic, which for some reason has a man tethered to it. And so what we're interested in is looking at that interaction between the binding site and the antibiotic. And so we have to really tease apart that architecture. And so fortunately, we can use X-ray crystallography and molecular modeling to actually look at how a bound Kanamycin-A looks like in that binding site of a protein. And I'm not going to go into too much detail with this, just to say that this is Kanamycin-A, and you're able to map all the interactions between groups in the binding site and the molecule. And that allows you to determine which positions on the molecule are important, either for binding or the positions where the modifications are taking place. And so we have that information for Kanamycin-A. And the position that we've been modifying is this one right here. Okay? And that's kind of the work that the biochemists do. And so what is the hypothesis then? In short, our hypothesis was that by substituting halogen atoms at key positions known to be targets of these aminoglycoside modifying enzymes, we could create a set of halogen modified aminoglycosides that will impart altered binding to that defense enzyme, so the possible resistance would disappear. Okay? And that would translate to having new aminoglycosides of antibiotic importance. Okay? And halogenation is something that we chose to do for several reasons. The first is it's been shown recently that substituting halides into positions on a molecule have dramatic effects on binding affinity. Okay? And so we started out being interested in making substitutions of all of the halides to Kanamycin-A. But another thing that's important is today, almost 1/3 of new drug candidates, and that's just not antibiotics, everything, are halogenated molecules. Because what's been found is having a halogenated molecule improves oral absorption. It facilitates crossing biological barriers and prolongs shelf life. So all of those things are important also. Okay? And so that brings us to what I actually did. I'm a synthetic person, and so biochemists designed where we should do the modifications and what we should, modify it with. And so then I was told, okay, here's kanamycin, make the compound. Okay? And carbohydrates give a big challenge synthetically. Because if you look at this structure, there are a lot of substituents on this molecule, OHs and NH2s. All of these have similar reactivity. Okay? And so you can't just say, I want to substitute something at this position and just throw a halide in there and expect it to end up at that spot. That's not how it works. So what we have to figure out is how can we direct the synthetic transformation to this position. And so even though they have relatively similar reactivities, they're slightly different. These NH2 groups are the most reactive on the molecule. So any reagent that we put in to do a reaction is going to first react with those groups. Okay? And so we need to do something to protect them so they won't interfere with the chemistry we do later. So what we use is called a protecting group. So we add something to this molecule that's going to react at these positions and then be inert to the other reactions that we do. Okay? And so this is the position we want to modify, but to do that we first have to protect those groups. Okay? And so to carry out that synthesis, the first thing we had to do, we bought Kanamycin-A, and it comes as a sulfate salt. Okay? So in order to do any chemistry on this, we need to remove that sulfate. And we did this with ion exchange chromatography, which is really a simple concept. You have beads in a column that have, in this case, a positive charge. This is DEA cellulose, diethylaminoethyl. It has a plus charge on it. That's what's demonstrated here. So the beads have a positive charge. If you have a solution of Kanamycin-A sulfate, when you pass it through the column, the sulfate, which has a minus charge, is attracted to the positive charge on the beads. The Kanamycin-A, however, has a positive charge. It doesn't interact at all with the beads. It comes straight through the column. Okay? So we're able to isolate the kanamycin free of the sulfate. And this slide kind of demonstrates this. This is the column. And we took the sulfate salt of kanamycin, ran it through the column, and we used 1.5 gram portions with 35 grams of the resin. And we were able to recover about 1.32 grams or 88% of this kanamycin. It was in an aqueous solution. We then freeze-dried it to remove the water, and then we had a powder that we could continue with the synthesis. Okay. Now the next thing we did, and I'm going to see if I can pull this off, to talk about NMR spectroscopy. Anytime you're doing synthetic work like this, you have to be able to check to make sure you have the compound at each step in the synthesis. And NMR is one way of doing that, nuclear magnetic resonance. It's the same technology that's used for MRIs. We're doing it. So one thing I always say to people is kind of imagine it's like doing an MRI on a molecule, is you get structural information about it. And basically how it works is certain atoms, their nucleus will generate a small magnetic field. And when you put it in the presence of a superconducting magnet, so a large magnetic field, those nuclei interact with that magnetic field in a little different way depending on what kind of chemical environment they're in. Okay, so if you're looking at carbon atoms, for example, if the carbons are bound to different substituents, they're affected by the magnet a little differently. And so then when you hit it with radio waves, they absorb at a certain frequency. So each one of these lines that you see is an individual carbon. And you notice that they're different because they're in different chemical environment. Okay? And so when we ran the C13 on the Kanamycin-A that we isolated, we were able to identify all of these peaks. And I labeled 2 just so you would believe me. This is the second carbon, and we know that it is represented by that line. 6' carbon represented by this line. And so seeing that, we count up the lines, and we know we in fact have the proper compound to continue the synthesis. Remember what we said we do in the synthesis is to first protect those 4 amino groups. Okay? And to do that, we use a common protecting group, butyl oxycarbonyl, and you react it as the anhydride. So we took this Kanamycin-A, treated it with Bach anhydride and water and DMSO at 60 degrees for 6 hours, and we obtained this product in 74% yield. Okay? So again, what do we have to do when we make that product? We have to test it by C13. And so again, what we're looking at is each individual line here, by the way, except these 3 that are solvent, each of these individual lines corresponds to a carbon in this molecule. And I noticed that one of the Bach groups, I actually showed it structurally instead of abbreviating, because I wanted to demonstrate when you add these groups to the molecule, you are adding these carbons and this carbon that is doubly bound to an oxygen. We know where those should show up on the spectrum. And just to illustrate, I've added 4 Bach groups. So I've added 4 of these C double bond O's, and that's where they are right there. 1, 2, 3, 4. So demonstrated that we have in fact made this. And just for fun, you can overlay the spectrum for Kanamycin-A, the starting material, and you can see that some of the other carbons are altered, but what we've added is the methyl groups here and those C double bond O carbons here. So we have in fact established that we have formed the proper product. Okay, then we now have the situation where we have these 4 groups protected, and remember we want to do chemistry here. Fortunately, this hydroxy group is more reactive than these others. And so we can now do chemistry specifically at that position. However, that OH group is not reactive enough towards, just directly adding a halide. We have to make it more reactive. And to do that, we reacted it with this triisopropyl benzyl sulfonate, abbreviated TIBS. And we substituted this group onto that position using the chloride in dry purity. And so that formed this TIBS compound. Reacted it for 70 hours. Yield was 50%. Okay, again, just so you believe me, we did do the C13 on it, and this is an overlay of Kanamycin-A, the Bach compound. Now this is the TIBS. And notice the changes. We see the isopropyl groups, those carbons that were off the benzene ring, and we also see aromatic protons, those are from the benzene ring, showing up. And so we've demonstrated that we have in fact produced that compound. Okay? And then we're at the point where we can try to put our halide in that position. Now this reaction we've only done once at 300 milligrams, and we got a mass recovery of 70%. And basically I say mass recovery instead of yield because we got a mass of 70%, but we don't know what all the mass is because it's impure. And so purification is in progress. And I have to say that when I was doing the synthesis, I realized why I stopped being a carbohydrate chemist, because this chemistry is really difficult. And one of the things that's really difficult is the purification. And so, our next step is to try to build up a larger amount of this product to then be able to work on ways to do a proper purification. Okay? And then the future step would be to remove the Bach protecting groups that were on the amino groups, and we would end up with this iodo Kanamycin-A. So a 6 modified Kanamycin-A with the iodine at that position. Once that was made, we would then test it. Fortunately, our collaborators have produced some of the chloro analog of Kanamycin-A. And so they were able to do some preliminary biological testing on this compound. And here's what they found, and I stress that this is preliminary. But what they tested is its actual binding. So remember, what we're trying to do is see if the chlorinated, in this case, Kanamycin-A, has less affinity for the enzyme, meaning it doesn't bind as well. So it's able to escape missile defense. Okay? And to do that, we looked at a value called KD, Dissociation Constant. And basically what it's looking at from an equilibrium standpoint is, the concentration of ligand, which in this case is the Kanamycin-A chloride, separate from the receptor, which is the enzyme, versus the concentration of bound, where the actual enzyme is bound with the antibiotic. And so what would you expect to see on these values? If the larger the KD is, the less affinity you have, because more of the concentration is separate. And so we tested this Kanamycin-A chloride with 2 different defense enzymes. We compared Kanamycin-A with the chloro derivative. And what we saw in these KD values is, maybe not significant, but a slightly less affinity for the enzyme. Okay, so conclusions. We have developed and optimized several synthetic steps towards several halogenated aminoglycosides. Once we form that iodo analog, that analog itself can be used to produce the other halogenated analog. So that's a very important compound to make. That's why we were trying to make it first. We did do some preliminary binding studies on the chlorinated Kanamycin-A. I did, in fact, create several new projects for research students, and the 2 undergraduates, Sarah Gwen Moore and Sarah Bowman, are going to present preliminary results of this at the American Chemical Society National Meeting in New Orleans. And so with that, I'm concluding and ready for questions. Yes?
Have they looked at the halogenated forms that they've made, and does it still interfere with the
Yeah, see, that's an important question, because that's the second binding thing we have to look at. No, that has not been looked at yet. Because she's asking about, we were looking at the binding to the defense enzyme, but there's also, it has to improve, or maybe not improve, but still be able to bind to the ribosome to be effective. So there are actually 2 binding events that we have to eventually look at. Yes?
From the minds of all former organic students, how long did it take to get percent yields that were that high?
Optimizing is a big thing. Some of you might be out there who did 4 reactions, but you have to optimize those reactions, and it takes a long time. When you show it on the screen, it's like, yeah, we did that reaction, but it takes a long time. You have to work with different solvents. But in our case, on this synthesis, the big problem was purification, because those carbohydrates are really polar, and normal purification techniques we use were very difficult. Yes, because the geometry is important, but your question goes to the stereospecificity of things is very important too. I mean, the molecular architecture in a binding site is very specific. So it would matter, the conformation. Yes?
I believe there's going to be any change if I do have to make these analyses, results are recorded, modified Kanamycin-A.
Yeah, that's something, those, what they're called X bonds. And that's something that's, relatively new and being investigated by medicinal chemists. But, fluorine, for example, is the most electronegative. It is the only one that really does hydrogen bonding. But the others do what's called X bonding, which is a kind of charge transfer bond. But, I don't know the answer to that. That's why we're looking at all of them. Yes?
Have you tried deprotecting the halogenated before purification? Does that help at all?
Well, that's a good suggestion, because then we might be able to go into something like reverse phase purification. No, we haven't tried that. But that might be something where we build up a good inventory of that protected compound. That might be something to look at. Sarah Williams and Sarah making notes too. Yes. Yes, we're at the ground floor of this. We want to modify all the halides at that 6th position of it. We have 3 or 4 other positions on the molecule that we want to modify, but that was, the low-hanging fruit. It's a little easier synthesis, and so we thought we'd do that one first and see if we were seeing any kind of body affinity change before we started working on it. Because, you took organic, so you're going to understand what I'm saying. That at that particular position, stereochemistry doesn't matter. But when you start making modifications in the ring, it gets difficult because you have different possibilities for axial equatorial substitution. It gets kind of complicated. Yeah, okay.
Does this, if you do this modification, does this change the packability of this drug?
I would imagine. I don't know.
Show me one more on the bottom.